Hello everyone, this is Abikesh Prasada Kumar Mahapatra, a PhD scholar in Pharmaceutical Sciences from OPJS University, Churu, Rajasthan, India. Today I am going to discuss about a research topic, garden Cress seed, Lepidium sativum, its the isolation, extraction and evaluation of mucilage, and we have to evaluate it as a pharmaceutical excipient, mainly as a disintegrant. Regarding the introduction of Lepidium sativum is known as halal in India. It's widely and popularly called as garden Cress seed. The different parts which is very useful and popularly used in India is seeds, root and leaves. This plant is very viable in India and its uses in India very in different Ayurvedic Unani formulations. The seed part is very much significant and it is highly resource and rich of mucilages, alkaloids and alkaloid semi-lipinoids such as A and B. The mucilage part is mainly a complex and heterogeneous in composition. The mucilage mainly used in different formulations, in pharmaceutical formulations like emulsions, stabilizers, gelling agents, also binders, disintegrants and solubilizers. Now in this research we are going to use its mucilage as a disintegrant activity. The main objective and scope of work is to isolate and extract the mucilage from the seed and its use as a pharmaceutical excipient, as a disintegrant in a pharmaceutical formulation. To evaluate its disintegrant activity, we have taken a tablet formulation, mainly a sublingual tablet dosage form, and we have taken a model drug, and we are evaluated it against a synthetic available disintegrant Agdisol. The main scope is also extended to evaluate how the inherent properties of the material that can be used as a disintegration pharmaceutical formulation. So you can see that this is the brief manufacturing process of extraction process of mucilage. In the domain, different extraction processes are available for. We have adopted a different process and we have proposed some modifications. The modifications mainly related to the adjustment of basic pH, water is to seed ratio in 30:1 ratio, soaking of seeds and boiling of seeds, refrigeration of seeds before precipitation of the mucilage, and also optimization of drying temperature. Now why these things are required, whether it is highly essential, and how these steps helping us to get a better yield. So the basic pH mainly help us to increase the yield by solubilizing or hydrolyzing the insoluble components, and the maintaining of a water to seed ratio is mainly based upon our this ratio by doing different experiments, and the boiling steps mainly help us to get the extraction process better, because the mucilage is present around the seed and it is little bit adhesive in nature. So the boiling steps may ensure to effective removal of the mucilage from the seeds. Apart from that, the drying temperature, it helps us to increase the yield by hydrolyze the polysaccharide which is present in the mucilage. These are the parameters we have proposed to get a better yield. Now regarding the manufacturing process, we can see that we are adopting a soaking process, filtration, then treatment with acetone, then formulation of coagulants, then precipitation, then dried mucilage. Here one step we have evaluated is the using of a hand blader, which is used to remove the mucilage present around the seed. You can see that this is the seed and this is the dried mucilage which is obtained after process, and this is the powdered mucilage which can be used as a disintegrant. Now this is the we have covered the extraction part. Now the main part, which is we can evaluate how you can evaluate as a disintegrant in pharmaceutical formulation. For this we have taken a formulation and we have chosen a sublingual tablet formulation. Then why sublingual tablet? Because in sublingual tablets or ODT formulations, the tablet need to disintegrate quickly or within few seconds or within 1 minute. So in this case, the role of a disintegrant is highly essential. So that's why to evaluate a disintegrant property of a disintegrant, this type of formulations gives us accurate and better result. So that's why we have chosen a sublingual tablet dosage form. And regarding the selection of excipients, we have taken beta CD along with SNRP in 1:1 ratio. This is based upon our study, and we have used beta CD to increase the solubility. And the starch we have used as a binder, and lactose monohydrate is a diluent, colloidal silicon dioxide is a glidant, mannitol to increase the mouthfeel effect because we are using in mouth and it's increase the patient compatibility compliance. And Lepidium, Sativum, seed mucilage, cross cramulose both are used as a disintegrant, aspartame as a sweetener, spearmint flavor we are using as a flavoring agent, and magnesium stearate as a lubricant. You can see that here AF24 is a formulation with a synthetic disintegrant Edisol, and 28, 29, 30. These three are different concentration of seed mucilage. Now, then again, why from where this formula comes from? Actually, we have optimized this formula by using a DOE experiment, and from the optimized formulation, we are using different concentration of seed mucilage to optimize its concentration. Now, regarding the manufacturing process, the manufacturing process actually mainly depend upon the direct compression process. In the direct compression process, we have used different excipients. We are directly mixing it for 15 minutes. Then you are adding the magnesium stearate for 2 minutes. Then you are using the fine blend for compression activity. Next is coming from the compatibility study, whether when we are using a different excipient, a plant extract, which is having different components, it is the high chances of incompatibility with the formulation. So to study the incompatibility, we have performed the DSC thermogram. From the diagram and the table, you can clearly identify that the DSC thermogram of SNRP remains same in the manufacturing formula. And there is no change, which is from 151.3 to 151.8. We can say that from this, we can say that the formulation is quite compatible with our excipients. And then again, further evaluation, we have done the excited study. In the excited thermogram, we can see that the characteristics of SNRP and characteristics of seed mucilage of Lepidium sativum is present in the final formulation. There is no disappearance of pricks, or there is no intensity change in intensity of peaks. This clarifies that our formulation is quite stable with the excipients taken. Then come to evaluation of pre-compression and post-compression parameters. The pre-compression parameters will be mainly evaluated, needs to evaluated because we are using a formulation where we are using is for direct compression process. And the direct compression process, we need that there is no, we are not taking any granulation process. The process mainly influenced by the excipients we use, and the excipients we have selected in our formulation mainly based upon the type of formulation and the choice of excipients. So to evaluate the flow properties and the compressibility index, we have done the pre-compression study. In the pre-compression study, we have evaluated the angle of repose, Carr's compressibility index, and Hausner's ratio. These properties clearly signifies that it is having good flow property and a high compressibility index. So all four formulations are satisfying our criteria for a direct compression process. Now, post-compression parameters. The post-compression parameters mainly depend, we have evaluated that weight variation, hardness, friability, which are the common parameters. Apart from that, three critical parameters for sublingual tablet dosage form like dispersion time, wetting time, and disintegration time. Here you can see that as we are seed mucilage, we are using as a disintegrant activity during harsh condition or during stability, it may change its property. So it's again need to be evaluated. So what we have done during we have exposed the product in stability, and we have checked its stability for up to 3 months. And in the initially, we can see that the results of the initial study, it can signify that all formulations having similar resolution profile. And in stability, you can see that there is no change in the properties. All properties, including the disintegration, wetting time, and dispersion time, all are same. And the release profile also, there is no change. From this, we can say that our product is quite stable in the formulation, and there is no inherent change, or there is no change in the properties of the effective disintegrant. Now, as we are seeing that this is having good disintegration lead to the disintegration activity. For evaluation of this activity, I want to highlight here that the parameters that responsible for this activity for this extracted mucilage is swelling index and swelling index. Apart from that, the water absorption capacity and the particle size of the disintegrant. So swelling index. So how swelling index is affected? Swelling index is the higher settling, means the settling volume. Higher settling volume requires more disintegration energy. And the lower settling volume liquid, less disintegration increases the disintegration time. It mainly works by swelling index. When it comes in contact with water, it forms a jelly, and it rapidly swells, and the matrix material comes outside, which is similar to the properties of some other disintegrant like explosive. This type of behavior mainly helps in disintegration activity, which leads to the wetting time and also the dispersion time. Another one property that is responsible is the water retention capacity or absorption capacity. The high water absorption capacity enable it to form as a gelling agent, and the carboxylic moiety present inside the mucilage helps to acquire more water when it comes in contact with any solvent. So this water retention capacity, high water absorption capacity, attracts water, and it helps in disintegration activity. The water absorption capacity and the settling volume mainly responsible for this activity. Apart from that, we can observe the particle size. The particle size, higher the particle size, more the disintegration activity. We are having higher particle size. This also leads to the more disintegration activity. Now come to the conclusion. From this study, we evaluated that it can be used as a disintegrant activity. The disintegrant activity shown by its higher particle size along with its water absorption capacity and also its settling volume. Even if as compared to the synthetic disintegrant, it's showing higher wetting time, dispersion time, and disintegration time. However, the dissolution time remains dissolution or in vitro dissolution is same for both the product. From this, we can say that we can use it as a disintegrant in different formulations, like rapid dissolution showing product like ODT and sublingual tablets. Apart from that, the extraction process we have used is very simple and cost effective, and the solvent used was acetone. From this, we can conclude that we can use it as a disintegrant in different pharmaceutical formulation and replace the synthetic disintegrants. And these are some references I want to use for this product. Thank you for your time. Now I am asking for any questions.